Good morning, everybody. My name is Daria Fresia. I am a PhD student at the Jules Garin Eye Hospital in Lausanne. And today I would like to present the results that I obtained so far in my PhD project, which is the role of ear stress in hypoglycemia-induced mouse retinal cell death. We decided to study hypoglycemia because it's well known that almost 100% of people affected by type 1 diabetes suffer from at least one episode of severe hypoglycemia per year. Hypoglycemia is more harmful in those tissues that have high glucose demand, such as the photoreceptor cells in retina. Therefore, we believe that the hypoglycemic consequences can contribute to the neurodegeneration of the retinal cells, and it is an event that can occur earlier than the microvascular complications. In this part of the project, we investigated the role of ear stress in hypoglycemic conditions, and to do that, we used two models, the 61W photoreceptors-like cells in vitro and the ear stress associated indicator mouse model in vivo. This is a transgenic mouse that carries the mRNA encoding for the human X-box protein binding protein 1 fused with an mRNA encoding for a GFP protein. So when the XBP1 undergoes splicing following an ear stress stimulus, we can easily detect the GFP signal. Talking about the results that we obtained in vitro, first of all, what we performed is that we cultured 61W photoreceptors-like cells for 24 hours in low glucose conditions, and we examined the expression of all the genes involved in the so-called unfolded protein response. So first of all, we detected the XBP1 splicing, and then we evaluated by qPCR the expression of the genes involved in the UPR. And as you can see, we have an increase in all the genes involved in the UPR, which is true also at protein level for all the genes that we tested. Next, we tried to evaluate if the ear stress may have a connection with a decrease in the antioxidant defense in the cells, namely with the glutathione content. So we administered butyonine sulfoxamine to 61W photoreceptors-like cells in two concentrations that are able to reduce GSH content in the cells of more than 90%, and then evaluated the ear stress gene expression. And as you can see here, we weren't able to detect any change in the expression of the ear stress genes, which means that ear stress is present, but may not have a link with the glutathione content in the cells. So moving on to the results that we obtained in vivo. First of all, we validated our model by tunicamycin, which is an ear stress inductor injections, both intraperitoneally and intravitreally. And as you can see here, we have an increase of the GFP signal both in liver and the retina, talking about the intraperitoneally injection, and also in the intravitreal injection we have an increase in the GFP expression. So what we performed next is that we performed a retinal explant. So we isolated the retina and cultured on filters for 48 hours in different glucose conditions, and as you can see here, at 1 millimolar we have augmentation of GFP signal in those structures that are likely Müller glial cells, which is interestingly similar to the result that we obtained in hyperglycemic conditions. Let's say 25 millimolar of glucose, which means that there is the beginning of the retinal degeneration at 1 millimolar concentration of glucose. So to come to the conclusion, even though the ear stress may not be linked with the decrease of the antioxidant defenses in the cells, ear stress is present after 24 hours of hypoglycemia, and it is present also after 48 hours of administration ex vivo in retinal explants. Thank you.